Hello everyone, I'm a first-year neuroscience master student from the University of Bordeaux in France. The topic I would like to explore in this video is the use of the CRISPR method on oligodendrocyte progenitors to allow the remyelination of neurons. Myelination is a key process in the constitution of neurons, in other words, neurogenesis. It allows action potentials, which are electrical signals located on the membrane of those neurons, to be conducted properly all along the axon, as you can see on this drawing. And it is because it is such an important and interesting topic that researchers from the University of Edinburgh decided to work on it. Laura Wagstaff and Anna Williams took a look at how CRISPR-edited human ES-derived oligodendrocyte progenitor cells improve remyelination in rodents. Let us take a look at what all of this means. Oligodendrocyte progenitors are stem cells that derive from embryos, here shortened as ES for embryonic stem cells. Their function is intimately linked to the process we previously discussed, neuron myelination, specifically in the central nervous system. Previous studies have shown that these progenitors are unfortunately subject to internal and external factors that can inhibit them if added without modification in the nervous system. So simply adding oligodendrocyte progenitors would not necessarily work in human bodies, or rodents for instance. We will now refer to those progenitor cells as HOPC, for human oligodendrocyte progenitor cells, to simplify the understanding. The aim of this study is to determine if there is a way we could potentially modify HOPC to avoid this issue and to improve their transplantation, and ultimately the myelination of neurons. The method that has been used, CRISPR-Cas9, is a relatively new technology that allows gene editing. The method is relatively simple. An RNA guide is used to target the specific sequence that we want to edit, and an endonuclease, Cas9, breaks the RNA to allow the editing to happen. To test the hypothesis that a modification through CRISPR of HOPC could improve myelination, they followed this protocol. The first step was to generate those modified HOPC. To do so in vitro, they first had to modify their precursor, the embryonic stem cells. They have chosen to delete NRP1, the receptor of a protein named SEMA3A. SEMA3A is a highly expressed protein in demyelinated neurons. This protein has what we call a chemorepulsive property. It repulses the HOPC away from the demyelinated neurons, making it more difficult to quote-unquote fix them. If we target this receptor, it could prevent the binding of those two components, and then prevent those effects. This is the enhancement we were looking for. The second step was to explore the consequences of this modification. They first observed the migration of HOPC and assessed the proportion of cells who moved towards or away from the lesion loaded with SCMA3. They then calculated the proportion of myelinated fibers with the enhanced HOPC and the ones who were not. This was a way to determine the effects on myelination. So now let's look at the results that they have found and the potential prospects that they could lead to. As you can see on the left, this Western blot indicates that those new enhanced HOPC contain no NRP1. The control test has been approved. The results of the second step are what I would like to draw your attention to. There is a clear and increased migration of enhanced HOPC and a clear repulsion of the ones who are not. As expected, the genetic modification allowed the first ones not to be affected by the chemorepulsive effects of SCMA3A and stay located on the desired area. What they found is that not only the migration is enhanced, but it also leads to improved remyelination. As you can see on this figure right here, the percentage of myelinated fibers has clearly increased, making this difference statistically significant. What we can take away from this study is that this genetic manipulation could overcome the issues that we faced with older methods. You may have heard of multiple sclerosis, a neurodegenerative disease in which we can observe the presence of neurons who have lost their usual myelin sheath due to inflammation. This process is called demyelination, and what it unfortunately causes is progressive disability. You can now see why this method is so interesting. It can allow a better transplantation of those cells and ultimately leads to a remyelination of affected neurons, on rodents at least. We, however, have to keep in mind that those results are still only preliminary. They are applicable to rodents, but may be more difficultly replicated in human bodies. As a final word, I would like to add that whilst we cannot be sure of it yet, this could still be a promising therapeutic option for multiple sclerosis patients if the transposition on human beings goes seamlessly. Only time will tell. Thank you for listening.